right. So you are very much welcome to today's session. And in today's session, we are going to consider a few common essays. And because according to our schedule, today we're going to have essays, and also tomorrow we're going to consider again a good number of essays. Um, Even today, around the next session will be in the evenings. So um, So basically, that's how we are going to move today. All right. So, as far as essays are concerned, you need to understand that you need to understand that essays can come from any topic as far as physiology is concerned. It can come from cellular physiology, nerve physiology, muscle physiology, blood physiology, where you have issues to do with blood clotting, the clotting factors. It can come from GIT, where you have things like uh, the swallowing reflex, defecation. It can come from the cardiovascular system. You can have renal physiology, endocrine, as well as respiratory physiology. So essays can come from any of those. And remember, um, according to your exam standard, you can only be given a maximum of 6 essays unless otherwise, maximum of 6 essays, and you will be required to only probably just write 4. And now you can now imagine the vast range of topics that you have, um, and the only to be given an opportunity to write 4 essays. Okay, so it's tricky. So you need to know all the major essays from each and every topic for you to be safe. Okay, are we good to go? Can we start now? All right, so let's um, begin. So um, this file, we already considered some of these topics, some of these essays, and they came in your test three. So we just ride on the protocols that we already established by us looking at us not looking at the at the essays that we already talked about. Okay, so we covered all these that I'm passing. We already talked about them. In case you are not there, please kindly watch the video. It is there both on our system and also on YouTube. You'll find it. Okay. All right. So we begin from question number 9. So this is the question that this is where we ended when we were revising for test 3. That is where we ended when we were revising for test 3. So we'll start from here. Okay, so this question is about the cardiac cycle. Okay, this question is about the cardiac cycle. So the question is requiring you and I to describe the physiology of the cardiac cycle. Okay, the physiology of the cardiac cycle. I believe by now you should be able to define what cardiac cycle is. All right. Are we safe with the meaning of the word cardiac cycle? Is everyone safe on that part? We know what cardiac or what the cardiac cycle is, right? So you can just give me a signal, either a thumb if everyone is okay, so that we just get straight into what we are uh, just describing the physiology of the cardiac cycle than us defining the terms. Is everyone comfortable? Okay, someone is saying no. Okay, so only one person, only we only have one person. All right. Okay, so let's begin from there. Let's begin from there. So before we can describe the physiology of this cardiac cycle, let's try to lay a foundation of what cardiac cycle is all about. Okay, so by definition, when we say the cardiac cycle, this is just basically the events. Okay, this is just has to do with the events that take place from the beginning of one heartbeat. Okay, from the beginning of one heartbeat to the beginning of the other heartbeat. So that period in between is what we call cardiac cycle. Okay, so you need to understand that each cycle is basically initiated by what we call the sinus node. Okay, by the sinus node, which is just located in the superior lateral way of the right atrium, basically just near the opening of the superior vena cava. Okay, so as far as cardiac cycle is concerned, we have what we call diastole and we have what we call systole. So diastole just has to do with the relaxation. Okay, so the relaxation in which the heart gets filled with blood. And then when you have systole just has to do with contraction. Okay, so contraction has to do with emptying of the heart. Okay, so another thing, even before we describe the physiology of the cardiac cycle, you need to understand that 80%, 80% of the blood, 80% of blood basically flows directly through the atria into the ventricles just before the atria contracts. Then you'll find that atrial contraction only causes an addition of 20% of blood into the ventricles. Are we together? We've gotten the uh, that part. Can we proceed now? All right. So the quicker you respond, the quicker we move. Okay. So now coming to our question, the question is saying describe the physiology of the cardiac cycle. So basically, when we talk about the cardiac cycle basically has when you want to describe the phases of I mean the physiology of the cardiac cycle, you will have to you will be you will be required to basically get to know the phases of the cardiac cycle because it is through those phases that you can completely describe the physiology of the cardiac cycle. Okay, so we have approximately, let me just say, we have about five phases. Okay, we have five phases of the cardiac cycle, and you need to know them. So the first phase is what we call atriosystole. Okay, we have what we call atriosystole. Okay, so under this phase, basically, atriosystole, the atria gets to contract. Okay, so when the atria gets to contract, it pumps blood into the ventricles. Okay, so under atriosystole, also the atrial pressure increases. Okay, the atrial pressure increases, and when the atrial pressure increases, you'll find that the mitral and the tricuspid valves will open. Okay, when they open, what will follow next is that blood will now flow into the ventricles. Then when blood is flowing into the ventricles, that is the beginning, that will be the beginning of ventricular filling. So that is the first thing as far as cardiac cycle is concerned. The next thing has to do with ventricular systole. So ventricular systole first phase, okay, ventricular systole first phase, which is termed as volumic contraction. Isovolumic contraction. So what happens in ventricular systole first phase is that the ventricles will basically contract, and when they contract, we'll have an increase in pressure. And when there is an increase in pressure, we'll have the mitral and the tricuspid valve closing. Okay, so when they close, you also have the aortic and the pulmonary valves are closed as well on this phase. Okay, so you'll find that the ventricular pressure will definitely tremendously increase or rise rapidly. Okay, so what happens under isovolumic contraction is that the volume will remain constant. Okay, so that is ventricular systole first phase. Then you also have the third one, which is ventricular systole, and now this one is second phase. Okay, ventricular systole second phase. So second phase has to do with ventricular ejection. Okay, so remember in the isovolumic contraction, the aortic and the pulmonary valves were closed. Now, in ventricular systole second phase, you'll find that the ventricular pressure will exceed the aortic and the pulmonary valve pressures. So, what will happen is that the aortic and the pulmonary valves will definitely open, and when they open, blood will be ejected into the aorta and the pulmonary artery. Okay, so now after blood has been ejected, definitely what you expect is that ventricular volume will definitely decrease. Okay, then the aortic and the pulmonary pressures peak will be this value right here. Okay, then on the fourth phase, what we have is ventricular diastole. Okay, we have ventricular diastole. So this diastole is early diastole. Okay, this is early diastole. So this is also known as isovolumic relaxation. Okay, all right. Someone is saying I'm too fast. All right, let me be. All right, so I was talking about that the fourth phase under cardiac cycle is what we call ventricular diastole. Okay, is this okay now? Okay, so we have what we call ventricular diastole. So this ventricular diastole, what happens? Remember, in the preamble, when we are trying to lay a foundation, we say that diastole has to do with relaxation. Okay, so this ventricular diastole, which is the early ventricular diastole, what happens is that the ventricles will relax, and when they relax, pressure decreases. Then the aortic and the pulmonary valves will definitely close. Okay, so while these aortic and pulmonary valves, which were open, close, the mitral and the tricuspid valves will still remain closed. Then from there, the ventricular pressure will drop. Okay, so when the ventricular pressure drops, volume will still remain constant. Okay, then from there, we have the last phase under cardiac cycle, which has to do with ventricular diastole. So this is ventricular diastole, but particularly late ventricular diastole. So what happens in late ventricular diastole This has to do with ventricular filling. Okay, this has to do with ventricular filling. Okay, so you'll find that in a nutshell, what happens in ventricular diastole is that the chambers of the heart and blood, I mean the chambers of the chambers will relax. Okay, the chambers will relax, and when they relax, blood fills the ventricles passively. Now, what happens in ventricular diastole? That was just a nutshell, a summary. So what happens in ventricular diastole is that the mitral and the tricuspid valves open. Okay, so when they open, blood flows into the ventricles. Then when blood flows into the ventricles, remember this is ventricular filling. Okay, so the volume of the ventricles will increase. Then the atrial pressure will in turn decrease. Okay, so what happens is ventricular pressure will remain constant. So by covering all what I have said, you would have answered the question: describe the physiology of the cardiac cycle. Is everything clear? Is there anywhere I need to reiterate or re-elaborate, or everything is clear? We can proceed to the next question. All right. Then we proceed to the next question. So question number 10 is about skeletal muscle contraction. So the key aspect here is to know that this has to do with skeletal muscle contraction. Okay. So the question says, explain the process of skeletal muscle contraction in humans. Explain the process of skeletal muscle contraction in humans. Okay. So this has to do with contraction basically in humans. So this is muscle physiology. Okay, this is muscle physiology. So now, under muscle physiology, remember, there are a lot of things that we talked about under muscle physiology. Just to remind you about the things that you talked about. So under muscle physiology, you covered the properties. Okay, you covered the properties of of muscle tissue. You you looked at the general functions of muscle tissue. You looked at the properties of the characteristics of skeletal, cardiac, and smooth muscles. Okay, you looked at all those. You looked at the skeletal muscle in detail, where you considered its structure and its functions. Okay, you covered all that. Then you also went ahead and talked about the sarcomere. Okay, talked about the sarcomere structure. From there, you talked about the neuromuscular junction. As far as skeletal muscle is concerned, you looked at the neuromuscular junction. Um, you talked about the excitation, contraction, coupling. Okay, you talked about that. That is also a very good essay. Excitation, contraction, coupling. A very good essay can come from that. Okay, you talked about the sliding filament. Okay, you talked about the sliding filament. Okay, Then I think you also talked about, you considered a few clinical correlates where you talked about myasthenia gravis, you talked about the Lambert-Eaton syndrome. Okay, I think you also talked about summation, tetanus. You talked about muscle twitch, things, all those things. Okay, but nevertheless, let's focus on this, the demand of this question. Okay, so remember to be specific in your writing as far as essays are concerned. Just get the main points. Okay, then it was emphasized during your assessment that many times you don't just need to start just boom from the clouds, expecting that the marker or the examiner will just guess whatever you want to talk about, but you need to just give a brief introduction to what you want to talk about. All right. So explain the process of skeletal muscle contraction in humans. Okay. So this is physiology, step by step. Okay. So, step number one, what we are going to consider has to do with neural stimulation. Okay, um, neural stimulation. So we are starting from the beginning until to the we experience the contraction. Okay, so you have neural stimulation. Okay, neural stimulation, which you will find that uh, when when a signal has been sent from the central nervous system, it will be transmitted to the muscle fiber. So what happens is that the neural signal, so that neural signal, in this case we're talking about the action potential. Okay, so the action potential will basically travel down the motor neuron. Okay, so it will travel down the motor neuron, and then when it reaches, it will reach the neuromuscular junction. Okay, so when it reaches the neuromuscular junction, then we have step two. Okay, so under step two, which has to do with neuromuscular transmission, what happens is that at the neuromuscular junction, you will discover that the action potential basically will trigger the release of neurotransmitters. So in this case, we'll consider acetylcholine. Okay, are we are we together? Are we together, people? Hopefully, I haven't lost anyone. Okay, someone is saying that I repeat. Okay, so I was saying that when, okay, someone can't see the screen. Are you all are you able to see the screen? Miss Zimba, kindly log out and log in. All right, okay, all right, okay. So now, I was saying that when the action potential, okay, that has been generated, so I haven't considered how the action potential has been generated, no. But here we're just trying to look at how skeletal muscle contraction occurs, okay? So we take it, the action potential has occurred, and now that action potential has traveled, okay? The action potential now um, would have basically traveled. Okay, Or if we can put it this way, remember I've said that it starts from it involves neural stimulation. Okay, it involves neural stimulation. So what will happen is, since neural stimulation now has to do with a signal from the central nervous system through a neuron, basically to the muscle fiber. So what will happen there is that you have an alpha motor neuron. Okay, you have an alpha motor neuron. Okay, that is where now step two comes in. You have an alpha motor neuron. So that alpha motor neuron is the one that supplies the muscle fibers. Okay, so what will happen is that um, when the acetylcholine has been released from the motor neuron, what will happen is that that acetylcholine or the neurotransmitter will basically bind to the receptors on the muscle fiber. So the name of those receptors are what we call the nicotinic receptors. Okay, nicotinic receptors are, that is where the acetylcholine will bind to. Okay, so when it has binded to the nicotinic receptors, what will happen is the gated channels will open. Okay, and when the gated channels open, we'll have an increased concentration of sodium, which will now enter the cell, and potassium ion will leave the cell. Okay, thereby you'll find that thereby generating what, an action potential on the muscle cell. Are we together so far? Are we together? All right, so that was on step two, which has to do with the neuromuscular transmission. Okay, so we won't go much in detail, but I'll just bring out um, the physiology. Okay, so for all the nitty gritties of muscle physiology, make sure to watch the video on muscle physiology on our system. It is there. Okay, so on step number three, okay, on step number three, now since we've started generating an action potential, what will happen in step number three? So step number three has to do with muscle fiber depolarization. Okay, now we're going to depolarize the muscle fiber. Okay, so what happens is that the action potential that has been generated will now depolarize the muscle fiber. Okay, so when it has depolarized, when it has started depolarizing the muscle fiber, what will happen is there will be an alteration in the electrical properties of the sarcolemma. Okay, are we together? So what happens is that this depolarization comes as a result when threshold has been attained. Okay, so when threshold basically has been attained, there will be an activation, okay, on the sodium voltage gated channels, okay, which will now allow sodium to get into the muscle cell. Okay, so you'll find that once we attain the highest level or the highest voltage of depolarization, which is positive 30 millivolts. Once that has been attained, okay, once that has been attained, what will happen is that, how can we put this? Once that has been reached, as a result of the bulky influx of sodium ions, the inactivation channels will now close the sodium gated channels. Okay, so from there, what will happen is that, since we are talking about alteration of the sarcolemma, okay, depolarization has taken place, and then after we've reached the highest voltage of positive millivolts, you discover that the inactivation channels will close the gated sodium channels. Okay, so after that, now, we have step number 4. So step number 4 has to do with excitation, contraction, coupling. Okay, so what will happen, um, is that, how am I going to put this? So what will happen is that that depolarization that we generated in step number 3 will now trigger the release of calcium ions from the sarcoplasmic reticulum. Okay, from the sarcoplasmic reticulum. Okay, so now what happens in between here for us to reach to at the release of calcium ions from the sarcoplasmic reticulum is of great importance. So, remember, on your muscle cell we have an invagination. Okay, we basically have an invagination on the sarcolemma. Okay, so let me just use something like this. So this is your sarcolemma. So as your sarcolemma is coming like that, you are going to have an invagination on the sarcolemma like that. Okay, so it is, so this invagination that you are seeing is what we call the transverse tubule, right? So what happens is that as the positive charge or as the positive voltage that we created in step number 3 will be moving along this sarcolemma. Okay, it will be moving along this sarcolemma. Let me just change um, the color for better illustration. So as the voltage will be moving along this sarcolemma like that, it will be moving like that, it will be moving on this sarcolemma. What will happen is it will find this invagination in the T tubule. Okay, so it will just follow this invagination. Okay, so as it will move in that invagination, what will happen is that along the T tubule, the positive charge or the positive voltage will meet a protein that we call dihydropyridine. Okay, dihydropyridine receptors. Okay, so those dihydropyridine receptors are voltage sensitive calcium channels. Okay, so those channels, those receptors will basically be stimulated. Okay, are we are we together? So now what will happen is that the sarcoplasmic reticulum basically is just, if I can use, just by the side to this transverse tubule like that. Even this side you have, so please don't mind my drawing, I'm just drawing roughly. Okay, so what happens is that that positive, you have the dihydropyridine receptors. So the dihydropyridine receptors are located just there. Okay, and also you can have another one somewhere there. So these are the dihydropyridine receptors. So the positive charge, as it is coming down there, it will meet these dihydropyridine receptors, and when it meets these dihydropyridine receptors, they will get activated. Okay, so when they get activated, what happens is that, remember, The sarcoplasmic reticulum is basically rich in calcium ions. Okay, so now what happens is that the sarcoplasmic reticulum basically has a particular protein that we call ryanodine receptor. Okay, ryanodine receptor. So the ryanodine receptor, basically the ryanodine receptor is on this sarcoplasmic reticulum. Okay, the one that I've put in blue. So this is your ryanodine receptor. So what will happen is that when these dihydropyridine receptors get activated, what they will do is that they will get hold of the ryanodine receptors. And when they get hold of the ryanodine receptors, is that they will pull, okay? So these red receptors will pull these blue receptors, okay? And when they pull the blue receptors, which are the ryanodine receptors, what will happen is that they will open the sarcoplasmic reticulum, and after opening the sarcoplasmic reticulum, calcium ions will be released. Are we together? All right, so calcium ions will be released. Okay, so we are done with this point number one under the excitation contraction coupling. So what will happen is that after the calcium ions have been released, what will happen is that the calcium ions will basically bind to troponin, specifically troponin C. Okay, specifically troponin C part. Hopefully we haven't lost anyone. Okay, so when it binds to troponin C part, what will happen is that um, when it has gotten bound to troponin C part, the troponin C part will then pull on the troponin T site. Okay, remember troponin has how many parts does troponin have? How many parts does troponin have? Yes. So troponin has three parts. You have the troponin C site for calcium ion binding. You have the troponin T site for the binding to tropomyosin. You also have troponin I site for binding of actin. Okay. So uh, we are talking about, where were we? All right, yeah. So we said that the calcium, when the calcium has been released, it will bind to troponin C. So in return, after that binding, we'll have the pulling on the troponin T site. Okay. So the troponin T site will then pull on the protein known as tropomyosin. Okay, that is how this tropomyosin comes in. We'll have tropomyosin. Okay, are we good? So when that has occurred, we'll have the shifting of the tropomyosin troponin complex. So that was step number four. Then step number five has to do with step number five has to do with the sliding filament theory. Okay, so basically, when you are writing your essay, you won't need to put all those subtopics, subheadings. I've just put them for the sake of explanation. Okay, for you, all you need to do is just put all the necessary data in your writing. Forget about the unnecessary addition of words. Just pick the main points. Are we together? So now, the sliding filament theory. So what will happen is that the shifted complex, okay, the shifted complex will basically expose the myosin binding sites. Okay, the myosin binding sites basically on the actin filament. Okay, so remember the actin filament has to do with thin filaments. Okay, so what will happen is that after tropomyosin has given way for myosin, the myosin will then pull the thin filament, okay, which has to do with actin filament. Okay, are we together? So from there, the myosin heads will then undergo a conformational change. Okay, so a conformational change which has to do with what we call the power stroke. So what that will do is that we are going to have the pulling of the actin filaments towards the center of the sarcomere. Is that clear? Are we together? All right. So after the myosin has pulled the thin filament toward the middle part of um, of the sarcomere, you will find that the goal is for the two actin filaments to overlap. Okay. So when the overlapping has taken place, when overlapping has basically taken place, you will discover that during contraction, what you need to understand that during contraction, thin filaments will slide over one another. Then, according to the structure of the sarcomere, you discover that the H zone will disappear. Okay, so please take note that myosin does not move. Okay, so myosin does not move, but instead it acts as a lever. Okay, so during contraction, the thick filament doesn't move. Okay, so as a result, the A band will always stay the same. Okay, then when you follow your structure of the sarcomere, the I band will decrease in size. So after that has occurred, the last step is muscle contraction. So what will happen is that the sliding of actin past myosin filaments will result in muscle contraction, and at the end the sarcomere will shorten. As a result, it will lead to muscle fiber contraction. By so doing, we are done with question number 10. Are we good? All right, then we proceed. Now let's the next question, question 11 says state the four types of passive transport mechanisms. Okay? So this has to do with passive transport mechanisms. Can I just have someone, even in the comment section, just list for us the four types of passive transport mechanisms? Passive transport mechanisms. Just mention any that you know. the four types of passive transport mechanisms. Okay, someone is saying active transport, simple diffusion, facilitated diffusion. Someone is saying diffusion, osmosis, simple diffusion, facilitated diffusion, osmosis. Okay, okay, so I've seen three submissions. Okay, someone has also included filtration, exocytosis, endocytosis, simple diffusion, facilitated diffusion. Okay, are those the only submissions that people have made? This is um, one of the first topics. Okay, someone has also added osmosis. All right. Okay, so by now we won't spend much time on transport explaining because we've explained this for several times. Okay, we've explained this kind of information several times during various sessions of our revision. Okay, all right. So now let's get to answer this. So basically, when we are talking about passive transport, when I'm talking about passive transport mechanisms, these are kind of mechanisms which basically um, allow substances to move across the cell membrane without um, without requiring any energy. Okay, so you discover that we have various examples of passive transport mechanisms. Okay, so what happens is that as far as these mechanisms are concerned, the passive ones, we have we have diffusion, right? So we have diffusion, and under diffusion, that is where we find simple diffusion and facilitated diffusion. Okay. Then we also have osmosis. Okay. We also have osmosis. Is that clear? Are we together? Okay. So now we will first talk about simple diffusion. Okay, so we have simple diffusion. So under simple diffusion, basically by definition, this is just the movement of particles from a region or an area of higher concentration to an area which has lower concentration. We've been talking about these things since high school, so it don't be a big deal. Okay, so what are the characteristics of simple diffusion? So simple diffusion, there is no involvement of energy. The components being transported move down their concentration gradient, okay, and there is no carrier protein under simple diffusion. Examples include oxygen transportation, carbon dioxide, and lipid soluble substances, for example, steroids. So that is simple diffusion. Then we have facilitated diffusion. Okay, we have facilitated diffusion. So by definition, facilitated diffusion is just the movement of particles through carrier proteins or channels. Are we together? So this facilitated diffusion is also known as carrier mediated diffusion. Okay, so what happens is that the characteristics, yes, we don't need energy, and also the transported materials, they move down the concentration gradient. But in simple diffusion, we didn't have the involvement of a carrier protein. But now in facilitated diffusion, we have the involvement of a carrier protein, okay, or a channel. So examples include the transportation of glucose, amino acids, and also ion channels. Um, okay, so that was facilitated diffusion. Then we have osmosis. Okay, so what is this osmosis? Okay, it as well, we started looking at osmosis since high school. Okay, so basically osmosis is just the net movement of water molecules through a selectively permeable membrane. Okay, selectively permeable membrane, basically from an area of high water potential to an area of low water potential. Okay, are we good? So now, what happens in osmosis is that yes, even osmosis is a passive kind of transport mechanism, as its characteristics include no involvement of energy. There is a movement down the concentration gradient, and osmosis basically helps in the regulation of fluid balance. Okay, so examples of osmosis, we have water absorption in the kidneys. Okay, as well as you are able to we talked about water movement into cells during hypotonic um, conditions. Okay, so we talked about these movements, the effects of hypotonic, hypertonic solutions on the cell. We talked about that as that involves osmosis. Okay, so and then we also have what we call filtration. Okay, we also have what we call filtration. So filtration basically this how this is just the movement of particles through a membrane under pressure. Okay, when we have pressure, all right, that's when we have filtration. Okay, so you discover that, in other words, what we are trying to say is that filtration is just the movement of water and solute molecules. Okay, that is generally across the cell membrane due to, it can be due to hydrostatic pressure. Okay, normally generated by the cardiovascular system. Okay, so you need to understand that as far as filtration is concerned, generally, depending on the size of the pores on the membrane, on the cell membrane, only solutes with a particular size may be able to pass through that membrane during filtration. Okay, so characteristics: no energy required, there is movement down the concentration gradient, and it requires pressure difference. Okay, so examples include blood filtration in the kidneys, okay, as well as fluid movement through capillary walls. So there is also what we call there's also what we call vesicular transport. Okay, there's what we call vesicular transport. So vesicular transport, that is where you also find things like exocytosis, endocytosis. Okay, under vesicular transport. Are we together? Okay, so that was question number 11. We proceed to question number 12. We got to question number 12. So question number 12 says, with the aid of a well-labeled diagram, compare and contrast between temporal and spatial summation. Compare and contrast between temporal and spatial summation. So um, we did this when we were looking at membrane potentials. Okay, when we're looking at membrane potentials, and I remember I am the one who taught um, this, and we say that as far as membrane potentials are concerned, the order through which you are supposed to look at the membrane potentials include, you have the resting membrane potential. Okay, you have the resting membrane potential, which is just the voltage difference across the cell membrane when a particular cell is at rest. Okay, and then from the resting membrane potential, we say that there are various um, um, there are various aspects or components which see to it that the resting membrane potential is in the range. Okay, and those things include things like the sodium potassium pump ATPase. You have the leaky potassium channels, you have the leaky sodium channels. Okay, then from the resting membrane potential, we went ahead and now you consider what we call graded potential. Okay, you consider what we call graded potential. Okay, so Graded potential just involves you taking the rest membrane potential to a threshold potential. Okay, that is graded potential. You are taking the rest membrane potential to a graded potential. Okay, so it is that I mean from resting membrane potential to threshold potential. So it is that threshold potential which will help in opening the voltage gated channels. Okay, so grading potential is not only about taking resting membrane potential to threshold. It can also imply you taking the resting membrane potential away from threshold level, okay, through what we call hyperpolarization. Okay, so now under the graded potential, for us to reach threshold, we need to have more of excitatory presynaptic potential compared to inhibitory postsynaptic potential. Okay, so you for us to reach threshold, we need more of excitatory postsynaptic potential compared to inhibitory postsynaptic potential. Okay, so now for us to achieve that particular graded potential, it can be achieved via two methods or two mechanisms. That now leads us to the requirements of this particular question. So for us to reach threshold through graded potential, we need or we can achieve that through two mechanisms or processes. We have what we call temporal summation and spatial summation. Has someone gotten how we've arrived to this question? Are we together? So I was trying to lay foundation so that you know how we have arrived to this spatial and temporal summation kind of question. Are we together? Can we now proceed to answering the question? Okay, so the question is requiring us to do that with the aid of a labeled diagram. Okay, so the first one that we're going to talk about has to do with temporal summation. So temporal summation, by definition, is just the integration of multiple, okay, the integration of multiple synaptic potentials over time, basically resulting in the generation of an action potential. Okay, so now what are the key features of temporal summation? So you are going to have multiple synaptic inputs arrive at the same time, at the same synapse. within a short period of time. Okay, and then each input basically generates a small excitatory postsynaptic potential. And then what will happen is that the excitatory postsynaptic potentials will summate, will add up, thereby increasing the membrane potential. Okay, so you'll find that if the threshold potential is now reached through temporal summation, an action potential will be generated. Okay, are we together? So this, with the aid of a diagram on temporal summation, what happens is, let me try to draw something for you people with the aid of a diagram. So what happens is that temporal summation involves one presynaptic neuron. Okay, one presynaptic neuron repeatedly stimulating one postsynaptic neuron in order for us to reach threshold. So with the help of a diagram, this is how we can just um, demonstrate temporal temporal summation. Please, these are neurons. Okay, these are neurons. So this is a this one here is a presynaptic neuron. So this presynaptic neuron is firing impulses. Okay, so this is temporal summation. One presynaptic neuron firing on one postsynaptic neuron. Okay, then we have spatial summation. So spatial summation, this one refers to the integration of multiple synaptic potentials from different synapses on the same what? On the same post synaptic neuron. Okay. In other words, spatial summation involves, let me say, it involves three presynaptic neurons firing simultaneously on a single postsynaptic neuron. Okay. So the key features of this type of summation is that we have multiple synaptic inputs arriving from different synapses on the same neuron. Okay. Then each input as well generates a small EPSP. Then the EPSPs from different synapses will add up or will summate, thereby increasing the membrane potential. And then when threshold has been reached as well, an action potential will be generated. Now let's come to the comparison. Okay, let's come to the comparison. So the comparison is that both temporal and spatial summation, they involve the integration of synaptic potentials. Number two, both mechanisms, they contribute to the generation of an action potential as long as threshold has been reached. And number three, both of them, that is spatial and temporal, they require the summation of excitatory post synaptic potentials to be greater than the inhibitory postsynaptic potentials in order to reach threshold potential. Okay. Then, when it comes to the contrasting aspects, when we consider time, temporal summation occurs over a short time period, while spatial summation occurs simultaneously. Okay, spatial occurs simultaneously because we have more than one firing on a single postsynaptic neuron. Okay, then when it comes to the synaptic inputs, temporal summation, temporal summation basically it involves inputs at the same synapse, okay, from one presynaptic neuron. But when you consider spatial summation, it involves inputs from different synapses or inputs from different neurons, not just one. Okay. Then when it comes to the integration process or mechanism, you find that the temporal summation integrates um, inputs over time, while spatial summation integrates inputs from different locations. So basically, that is it on question number 12. Are we together? Can we move to the next question? Can we move to the next question? All right, then we proceed now. Okay, so now we go to question number 13. Question number 13 is about the sodium potassium ATPase pump. Sodium potassium ATPase pump. So the question is, explain the physiological importance. Explain the physiological importance. Okay, so we're not talking about how the sodium potassium pump operates, or but we have to give a background, of course. But the key is, what relevance does it hold physiologically when it comes to other systems, or when it comes to the operation of the human body? Okay, so the sodium potassium pump, which is just basically a very significant cellular mechanism, and this usually helps in the maintenance of ions, okay, and electrical potentials across the cell membrane. Okay, so the functions, what are the functions? So you discover that the sodium potassium ATPase pump helps in maintaining membrane, the resting membrane potential. Remember, I mentioned in the previous question that we have the resting membrane potential, and sodium potassium pump helps in maintaining that resting membrane potential. Okay, so how does it help in maintaining that resting membrane potential? It does that by pumping three sodium ions out and two potassium ions into the cell. Okay, that is how it works. Okay, because please, you need to understand that this sodium potassium ATPase pump, it has two receptor sites for binding potassium on the outside, and it has three sites for binding sodium on the portion of the protein that protrudes inside of the cell. Okay, so that is the first function. The second function is that this particular uh, component helps in regulating ion balance. Okay, so you'll find that this pump helps us to maintain um, the optimal intracellular and extracellular concentrations of sodium and potassium ions. Okay, and apart from that, it helps in volume regulation. It helps in volume regulation. Okay, because remember, sodium is one of the key players in regulating the volume of fluids. Okay, because when sodium leaves, it creates room for water to also move, okay, across cells. Okay, so that is the function of the sodium potassium ATPase pump. Then what is the physiological importance? Okay, number one, it is very important to the physiological function of the nerve tissue. Okay, so you'll find that the sodium potassium ATPase pump enables nerve impulses, okay, enables nerve impulses by maintaining the resting membrane potential and regulating the action potential generation. Then, secondly, it is also involved in muscle contraction. Okay, so you discover that this very pump helps when it comes to regulating muscle contraction and relaxation um, through the controlling of the availability of sodium and potassium ions. Then number three, it is also important physiologically when it comes to the function of the cardiac. Okay, when it comes to cardiac function. Okay, so you'll find that these same sodium and potassium ions, they help maintain proper heart rhythm and contraction force. Okay, that is specifically or detailed very well when you are talking about the cardiovascular system. Okay, so when you are considering the cardiovascular system, that is where these have the how these particular ions definitely come into play as far as the cardiovascular system is concerned. Okay, then apart from that, it is also important physiologically when it comes to epithelial transport. Okay, so you'll find that this very pump helps when it comes to regulation of ion transport across the epithelial cells in the kidneys, the gut, and other tissues. By doing so, you are done answering that particular question. Can we proceed to the next question? Can we go to question 14, which has to do with surfactant and surface tension? Okay, so I just give us 2 minutes break, just breathe, then we continue.